Our second speaker is Barbora Kozlikova, and Barbora is an associate professor at the Masaryk University in Czech Republic. Now she is leading a visualization research group. The research group investigates several visualization and visual analytics approach of biomolecular structures with specific focus on molecular dynamic simulations, molecular docking, and molecular interactions. The group also received several honorable mention awards at IEEE, and Barbora also involved very much in the organization of conferences such as BioMedVis, BCP, and FacilityVis. So we will have very nice talk from Barbora, and let's welcome Barbora, and the floor is yours.
Well, thanks a lot, Yun, for your very nice introduction. I'll start sharing my screen, hopefully. Okay, so I hope everything is working right now.
Yes.
So thank you once again. And what I'll be focusing on within my talk, it will be of course, something time-oriented, as the suggestion of, or the title of this session suggests. But I'll be focusing more on this biological part of our seminar, of our spring school, because as Yun was already mentioning, like this molecular visualization is deeply rooted in myself, in my heart. So that's why I'll be focusing on this within my presentation. So let's have a look together on the visualization of molecular data from the time varying point of view. And I will start immediately with showing you what I'll be talking about, basically. So here you can see a short animation of the movement of some protein molecule. And what's actually visible here is that you can see that there are some certain parts moving a bit faster, some a bit slower. You can see even some small molecules floating around the big protein, which are small molecules of water. And I'm just mentioning them because I'll be talking about them a bit later. So you just remember what I'm talking about over there. So this is a traditional manner how the biochemists are actually observing the animation, the movements of the molecular simulations. But let's start with like why these movements are important. So I'll be focusing mostly on something which is called molecular dynamics. And as you've seen in this video, it captures the movements of individual particles. In this case, these were atoms or some small molecules like these water ones. And it captures the molecular behavior over time, and also the interactions of this protein structure with some other molecules. This can be these waters which are floating around, or it can be some smaller molecule of ligand, for example, which enters the protein, and then some chemical reaction between these two molecules can undergo. So this molecular dynamics and these interactions between molecules are very crucial in many research domains related to molecular visualization. I'm just naming here those which I'm interested the most in, and these are drug design and protein engineering. Just one more slide to somehow defend the importance of studying molecular dynamics, because actually you can still watch just a static snapshot of a molecule and you can still see get quite a lot of information out of that. But this function is something which really adds quite a lot to the understanding of how the proteins and molecules in general are actually working. And I just put here the link to one quite interesting paper. It was actually for me quite outstanding result, which was published in 2012 by Hansen and his colleagues. And they are claiming that the similarity between proteins and their function is basically defined by their sequence, which means that we are having the constitution of the protein. We are having a sequence of amino acids which the protein consists of, then the structure, which means the 3D representation of the protein in space, and dynamics. So this dynamic part is very important in understanding what's the actual role of such a protein. So what is the challenge here? Why I'm even talking about this? The thing is that if we are observing some interactions of protein with some other molecules, this can take actually quite a lot of time. It's not because we can afford this. It's just because some procedures in cells, for example, they really require a certain amount of time to happen. So what the consequence of this is that if you would like to capture such a movement, such interaction, then we are usually capturing molecular dynamic simulations that are consisting of hundreds of thousands of time frames, which we have to somehow proceed afterwards. It can be even millions nowadays. And from the computational point of view, it's not a problem anymore, and we can easily capture this. But how this should be analyzed. So should we use, or could we even use such an animation, which I was showing at the very beginning? Actually, even Honza Biška in his presentation already on Monday, he was showing these nice numbers that imagine that we are having this 100,000 time frames, and if we are playbacking them in 1 frame per second speed, then it would take approximately 28 hours just to watch such an animation. And honestly, this is scary. And you really don't want the biochemists to look like this, and you would like to aid them somehow. So what's the current situation right now is that the biochemists are basically forced to shift from this traditional 3D representation and these animations to something more simplified. And this is the statistical analysis, as we can see here in this image. So basically from the 3D representation, they are trying to extract some properties, some parameters, which are somehow describing some interesting events happening within the whole simulation. And from these simple graphs, they are trying to extract what's important and what could be the crucial part of the simulation where the biochemist should focus on. And for example, here in this graph, you can see some distance measurement between two structures. And for example, here we are having some peaks in the distance. So this probably means that the biochemist should focus on this particular part of the simulation where something interesting is happening. They are getting closer to each other. But what's the problem here? The spatial representation is completely lost. Like they don't have the depiction of these distances, mutual orientation of amino acids, and so on. And it's very crucial to for really understanding the function itself. So you are losing this with this simple line chart, let's say. And another thing is that it took some time, but the biochemists already for decades are really well established and very well understand the spatial representations which are or have been designed within the last decades. And even some of them are really very abstractive ones. Still, they understand quite well how this is organized and how, for example, in this case, the big protein is accepting this small molecule of ligand, how the ligand is docking into the surface of the protein and so on. And this is very hard to understand and capture from simple graph. So how to help them to get back to this connection of having the animation or having the 3D representation still back and having the option to observe this, but still analyze such long simulations. So I will mention two basic approaches here. One is to aggregate. So we are just basically taking the whole simulation and extracting some information which we squeeze together, abstract from the temporal aspect of the data set, and showing everything at once. What are the benefits of such a solution is that first, you can see the overview of the whole simulation in one image, if it is possible. Of course, we can discuss how beneficial this is when we are having really long simulations. It really depends on what we are showing, what we are aggregating there, and how cluttered the visual representation after this can be. But basically, the overview of the representation of the simulation is a really nice aspect of this aggregation approach. Another is that if we are talking about 3D representation of the structure, within this aggregation, you can quite easily spot the spatial location within the protein, for example, where this interesting event, something aggregated, happened. So you are having this connection with the 3D structure again, and you can have information like this quite easily accessible. But of course, there are some drawbacks. One of them I already mentioned is that it can quickly become very cluttered, and I will show you some example later of this. So this is one of the issues which you have to keep in mind when designing some aggregation of molecular dynamic simulation. And another issue is that it's hard or sometimes even impossible to read out when the interesting events happen, because we completely omitted time here. So you can use, for example, coloring for conveying if this event happened earlier or later, but sometimes you cannot use the color channel because it's used for something different already. So it's really not so easy to spot here the temporal aspect. Another option is to navigate the user. So keep the time in your data set and guide the user to some interesting parts of the simulation and animate only those. So really keeping the animation aspect still there, but only use it for something which is really meaningful to watch, and the rest can be omitted. Again, benefits of that. We can observe the molecular dynamic simulation, I call it here like in this good old manner. So we can really see the animation of the movements, which is very handy and useful for the biochemists. We don't have this information loss, which was caused by the transfer to these simple graph representations. But of course, drawbacks as well. It requires quite decent and reliable way to actually detect what is the interesting event there. Because you can navigate the user to something which is meaningful, but this requires some calculation of, or like extraction of what is meaningful. So I was mentioning this already a couple of times. So what could be such an interesting event within the MD simulations? So I can give you a couple of examples here. So I would define an interesting event as something like a significant change in one of some geometric or physical chemical parameters or properties. And these are the good candidates for such an interesting event. And like one of the geometric examples could be, for example, some significant positional changes which are happening within the protein. So for example, in this image, we can see a protein consisting of two domains, and they are connected by this hinge here. And this hinge is causing some closing and opening of these domains. And for example, if the domains are opened, then it can signify that maybe some small molecule can enter this space, and then can be some chemical reaction, which is not possible when these domains are closed. So when the hinge is opening, this could be detected as a significant, interesting event within the whole simulation. Another example could be the speed of the reaction between protein and some other molecule. So if we are observing, capturing some interaction between two molecules within the MT simulation, then the speed of how fast these molecules are getting to the actual reaction is another significant property which we would like to observe. Another could be, for example, observe some peaks in the energy of the interaction between these molecules, and so on and so on. So there could be actually many more parameters, many more descriptors of such manner which we can consider, and the biochemists are still in this process of trying to find out which of them are actually important and interesting. But for that, it's like a chicken and egg, so they still need to have like the visual representation somehow to understand what's actually important and interesting for them. And one important thing is the combination of these parameters is something which is super interesting and important, because if you imagine that you are having quite like a dozen of these parameters, and each of them has some peaks in their representations. And if we just sum this together, then we can see many peaks in the MD simulation. And at the end, we can have, again, hundreds of possible interesting events there, which is not meaningful. So actually, for the biochemists, it's really interesting how to combine these parameters together, how they are influencing each other within the simulation, and how we can really correctly specify what's an interesting event with respect to all these parameters, let's say. Now I will show you a few examples of how these methods for aggregation could look like from the past. So I will start with one example from 2013, where basically, this is the information about the protein flexibility, which is aggregated to this single representation when the protein is visualized as a tube, and this tube is modulated in those parts, it's thickened in those parts of the protein, which is more flexible within the MD simulations. And the same information is actually encoded in color here. So the red parts are more flexible than the blue ones. Another example is coming from normal mode analysis. It's slightly different approach than molecular dynamic simulations. Jan Biška was also briefly talking about this in his talk, so I won't go into much detail about the technique itself, rather how to visualize this. So one of the typical examples of this is that basically the normal mode analysis is capturing some higher level movements of protein domains, some more abstracted parts, not individual atoms here. And the typical example how to visualize these movements is using some arrows, small glyphs like here. So this basically, the direction and size of these arrows is signifying in which direction within the dynamics these domains are moving and how fast they are moving as well. And it's the same situation on the right side here. Another example are visualizing of trajectories, which is something which probably you would imagine when I was talking about aggregation, that if I'm having some movement of some particles, then showing the trajectory is quite natural way. Again, it really depends on the data set, because sometimes it can really become very cluttered, as you will see later. But basically here I'm showing two examples where this was successfully applied. One of them is from Mathieu Chavin and his colleagues. And it's here on the left side where we are showing streamlines of lipid diffusion in molecular dynamic simulations on some large membrane systems. So basically we are seeing trajectories of these individual molecules, small yellow ones, over time. So you can see nicely the flow of them over time. On the right side, we can see an example from Tom Ertl and his colleagues from 2014, where we are having a vector field, and interesting regions in the vector field are basically visualized using these streamlines. And we are also having some arrows here in these parts, also here in the zoom view. And these arrows are encoding some detailed analysis of velocity of these particles here. And here is the example I promised at the very beginning with the water molecules. It was a project which we were collaborating with the guys from TU Wien in 2017. And we were observing trajectories of water molecules and how they are interacting with the protein. And already for just a couple of the trajectories aggregated together, we can have something like this. So such a hairball probably doesn't help anyone, even the biochemist, to understand what's going on there. And in fact, what happened is that only a very small subset of these trajectories was actually important and interesting for the biochemist, because most of the waters never entered the inner part of the protein. The protein, by the way, is hidden somewhere here in the hairball. It's not visible even. So only some trajectories were important and interesting for the biochemists. But even here you can see that this representation doesn't tell us much. It's still very cluttered. So what we did within this aggregation approach is that we were replacing these individual trajectories by isosurfaces, like here, which are basically signifying the space occupancy by these trajectories over time. So for example, in this red part here, somewhere deep inside, there were a lot of trajectories or a lot of molecules staying for quite significant portion of time. So that was the meaning of that. And if you imagine that you are having it for the whole protein, then you can have a very nice overview of the occupancy of space over time by these water molecules. I will now briefly jump to the second part, which is the navigation. So we are not omitting time this time. And what we are doing here is that we are trying to change the animation speed for these important events. So again, we have to have some meaningful way for extracting these parameters which are describing the important events. So we are starting with the molecular dynamic simulation. We are extracting and combining these individual properties, parameters together to something which we call a descriptive function. And this function is basically, again, like a line chart, is guiding the user from the spatial and temporal point of view as well. So from the spatial point of view, if there is something, some peak, something interesting in this descriptive function, then we are locating where this important thing is happening in the protein. And then this important part is shown in more detail, and the rest of the molecule is visualized in a very abstracted way. So this is basically how it looks like afterwards. So the important event is happening in this particular part in the protein. And moreover, we are using this information also for influencing the playback speed of the simulation itself. And I have this in this image here. Oh, sorry, it should be a video. So I'll just stop here. Yes. So here you can see that when reaching a peak in this descriptive function, the animation is slowed down. So you can really observe way better what's happening there, how this small guy, the ligand, is actually interacting with the amino acids around and so on. And of course, in this case, you can argue that we didn't solve the problem of not watching the whole simulation. But of course, the user can set it in that way when there's something or nothing is happening, like I just use this one, like in this area, then we can completely skip the animation. So that's how we can really squeeze the animation time for the user. Another approach which we were taking with colleagues from Barcelona in 2019 was basically, again, focusing on exploration of the interaction of protein with small molecules of ligands. And this time it was a simulation where we had one protein and in this particular case, 3 ligands entering in different times the inner part of the protein. And we wanted to observe when this is happening, how far from each other these ligands actually are, how far they are from the protein itself, and so on. So for each of these ligands, we were having one plot here. And again, these lines are different properties, parameters, which we were observing. And this background gray rectangles here are navigating the user to something which is important and interesting to observe. So for example, here we can see that this bottom part for this ligand, it was important for some reason. The reason we are not getting into much detail here, but for example, it got closer to the reaction site, so the user should really focus on this particular moment. What's also beneficial here is that you can observe what happened with the other ligands in that particular time spot when you are interested in. So that was another approach which we took. And I want to mention the last example, which is from Robin Skonberg and his colleagues from 2018. And basically, they were presenting via MD tool, which is a tool which integrates the statistics, some plots showing these individual properties which are measured, and the 3D visualization of the interacting molecules. In this case, they are having a big tubular structure here and a set of some ligands floating around. And when they are approaching to the tube, then they are somehow changing their appearance. Their angles are changing, so they are becoming more flat. And all this could be considered as one of the nice descriptors of an important event here. Moreover, what you can see here is that they were also tracing the trajectory of some ligand over time. So it also integrates some aggregation technique here, which we were mentioning before. So is everything solved? Because it looks like we are having quite nice approaches to this. Of course, not really, otherwise I wouldn't be here probably. So what's still the problem? It is still very complicated for the biochemists to understand how these individual properties which we are measuring are interplaying together. So what I was already mentioning, how to combine them in a meaningful way to really detect something which is important to observe within the simulation. So what we are doing right now is that we are trying to create some visual interface which will help the biochemist to intuitively design this importance function, which I was showing on a couple of examples, and to really understand how the different tweaking setting of the parameters influences the importance of these particular moments over time. So let's see, maybe some paper is coming out of that. So to summarize my talk, so I hope that I managed to convince you that the dynamic molecular movements are important for the biochemist, and it's really meaningful to focus on them from the visualization point of view. That it's very challenging to detect, visually depict, and also navigate the user to these important parts of the simulation, which is really meaningful to observe in particular detail. And one thing which I think, yeah, it's a buzzword which is everywhere right now, that everyone wants to use machine learning, but I still believe that in this case, it could really help, because once we are able to somehow define automatically what is important, at least to a certain extent, I think that we could try to train some neural network to help us with the analysis of these different parameters and how they are playing together. So I hope that this is quite big space for machine learning here as well. But let's see, we didn't touch this yet at all. So that was it from my side. Thanks a lot for your attention, and I'll be happy to take any questions.
So thank you, Barbara. We have the questions from Laura Garrison. The question is, have you explored or has there been interest from your collaborator in visualizing quantum behavior that enable molecular dynamics? For example, electrons teleporting to lower an energy peak for a reaction.
Yeah, thanks a lot for this question. Actually, there's a lot of stuff which is available also for observing interactions between molecules. Like I was showing really like a small subset of that, unfortunately, but there are techniques which can observe this both from the static point of view, but also like to simulate the animation or the interaction between these molecules within the animation. And you can see how they are interacting, showing the forces, showing what's going on there. But this is the field which definitely also requires a bit more attention, because you can show way more. And it also depends on how much information you can calculate from the biochemical point of view, because the geometry is not an issue. We can quite easily define the distances, whatever, calculate, it's not a problem. But to really calculate at least a decently realistic forces and everything which is what's happening there, and to visualize this properly and to understand again the interplay of these, I think this is still very challenging.
Thank you very much. I think right now we don't have further questions, but someone is typing, so maybe still wait for a while. Oh, okay, no. Okay, now I get it. So we have a question from Roden Den Luo. The question is, I wonder, do you feel and how do you handle the balance between visualization task and bioinformatic analysis task?
It's really tough, because to really focusing on something where you really would like to have the visual representation at least to as much extent as possible, also not just showing stuff, but also showing something which is meaningful and really, let's say, biologically relevant. This is an issue. So I think we cannot live without each other. So the visualization and bioinformatics, these parts are really playing together. In bioinformatics, at least from my point of view, I see it more about the analysis and calculation of different properties. It could be, for example, molecular docking, how two molecules are docked, how they are interacting together. So this is something which needs to be calculated. It cannot be just visualized. So we need to have some data behind, which are calculated using some computational tool. So I would see this like we cannot live without. we have to be together somehow, but really focusing on both parts is also very challenging. So but I can discuss this with you afterwards, maybe the social event, if you are interested a bit more about this, because before diving into visualization of molecules, I also spent some decent amount of time in the analysis of proteins themselves using some computational geometry approaches and so on. So, but there's no space for about talking about this right now.
Okay, I think we also have a quick question, so maybe I will ask it quickly. Does your approach also work for the protein-protein interactions?
Very nice question. We were also working on protein-protein interactions. I didn't include those here to the talk because I didn't have time and space for that. But again, if you would like to talk about protein-protein interactions and how to visualize them a bit more, we can do this afterwards on Discord, or we can jump into some table at the social event for this. Basically, it could work, but I think it would require a certain amount of tweaking there, because it's still challenging, for at least from my point of view, to get the proper simulations of two or even more proteins interacting together. And maybe it also requires different information extracted out of that, because when two proteins are interacting, then you are having the contact zone, you are having some amino acids interacting, and you have to somehow decide what's important there, how to assemble them together, and so on. So it's a bit complicated, and slightly different tasks have to be solved there, but we can definitely discuss this afterwards if you want.
Great, thank you, Barbara. I think we are running out of time, so I would like to close this session, and we will have a short break until 15:15, and we will welcome our next excellent speaker. So see you later.